Good morning, everyone. Thank you for joining this webinar. Today we'll be talking about the origins of life and how it met microfluidics. I'm Camila, and I'm a PhD student from the ProtoMet Consortium. This consortium is part of the Horizons 2020 projects from the European Union and part of the Marie Curie actions as well. The goal of this consortium is to better understand how life as we know it came to be. And we here at Elveflow are joining forces and lending our expertise in microfluidics to help them achieve that goal. So before we travel in time, I want to talk a little bit about myself. So I'm Brazilian, and I have a hybrid background in science and business. I am a biotech engineer from the State University of São Paulo in Brazil, and I have a master's degree in bioscience enterprises from the University of Cambridge. But most of my career was in business innovation and digital startups. And in a more personal note, I'm a global trotter. I visit 26 countries and I lived in 5 so far. So Brazil, US, the Netherlands, the UK, and now France. And I'm a dance lover. So I do tons of different rhythms. And here are just examples of bolero and tango, but there's quite a few others that I dance as well and I lived. So now, we can go back in time a little bit and think about the origins of life. When people usually think about the origins of life, they think about the primordial soup. That is that idea that the building blocks of life were made in this aqueous solution in the primitive earth with these very hard conditions. And this idea, or this hypothesis, was shown to be possible by a very famous experiment done by Stanley Miller in 1963. What he did, basically, he put together a few basic gases, let's say. So here we have water vapor, methane, ammonia, hydrogen, under harsh conditions. So he discharged electrical sparks to mimic potential thunderstorms of the early Earth. And he was able to collect some very interesting molecules afterwards. So he was able to collect glycine and alanine, which are amino acids, the building blocks of the proteins today. And here we have this extract from his paper, which I find super cool. He did a paper chromatography back then, and he could identify a few compounds that are organic, made from inorganic compounds, which is very interesting. And this experiment was redone in recent years by quite a few independent groups, and they were able to reproduce it and also find way more molecules than he was able to find back then because we had so many advances in the analytical techniques. So that's a good indicator of how life came to be. But there's quite a long way between having these floating molecules in this sort of soup-like environment to what we know as life. So how did we go from this primordial soup to the highly complex and organized structure of the modern cell, for example? Or even, if we don't think about the modern cell, but like a simpler structure that resembles a cell, how do we went from chemistry to biology? That's the big question that researchers in the origins of life field are trying to solve. And there are a few ways that people are trying to tackle this problem and fill this gap. The first one is what I call molecular systems. Basically is the idea of reproducing metabolic processes. So here, for example, we have the Krebs cycle. So researchers are trying to reproduce those biological processes using prebiotic molecules. For example, in this experiment specifically, they substitute the enzymes needed to do the Krebs cycle by metals. We know that, well, we believe that metals were abundant in the early Earth, and they are still very important parts of the enzymes that we have today, of modern enzymes. So these researchers from Joseph Moran's lab in Strasbourg, they wanted to see if only the metals would be possible to act as catalysts to make this reaction happen. So they managed to redo a few of the sequences of the reverse Krebs cycle using only metals, showing that yes, metals could be used as catalysts, and that might be how the first metabolic pathways came to be. And there's another approach that is compartmentalization. So in this sense, researchers are studying how the first compartments came to be, how they first built themselves. And compartments are fairly important because they concentrate reactions and they isolate reactions, providing potentially advantages to these systems in comparison to the environment. But before we go in more depth about how we study the origins of life, we actually need to define how we see life, what is life? And there is no consensus in the academic community whatsoever. So we're going to borrow one definition from Monard and Peter Ward from 2015, which fits really well with the goal of this webinar. So bear with me. To define life, we need to make a few assumptions. So first, we need to think of water as the universal solution for all life chemical reactions in living systems. Then we need to think about cells as the basic unit of all life forms, regardless of the complexity. The triad DNA, RNA, and protein is the basis of all life processes as well. And these life processes are made with complex molecules, which can be modified and transformed in a very dynamic and controlled way. And finally, we can see communication between the internal environment of this unit with the external environment and with different units. So taking these 5 assumptions into account, we can conclude that life, the emergence of life, is tightly linked with the emergence of dynamic compartmentalized chemical systems. What that means is, if we didn't have any compartments in the early Earth, life would not be able to exist. We would still be in the soup state, let's say. So compartments are a defining moment in the definition of life, in the definition of the emergence of life, let's say. And there are a few ways that we can study compartments. So first, there's the top-down approach to make compartments. So we take a modern cell, regular cell that we have access to today, and we strip it of all non-essential, or of all of the things that we think are non-essential, and create the minimal cell and see if that resembles what we think of as life. Or we can have the bottom-up approach, where we take the building blocks of organic, we take the organic compounds and building blocks of what we think make life, and we put them together to assemble the minimum possible cell, let's say, and the minimum possible thing that resembles life. Let's put it like that. So basically, the bottom-up approach is building the cell from scratch. And that's where life, or the origins of life, meet microfluidics. We can use droplet microfluidics to do templates of compartments. So first, what is droplet microfluidics? It's using the microfluidic channels and the immiscibility of liquids to do tiny droplets in a very controlled way. So here we have a few examples. The T-junction and the cross-junction are examples of droplet-making devices that use shear force to break the aqueous phase into tiny droplets with the force of the oil phase, for example, in the case of water in oil droplets. And we also have the glass capillaries, which are more 3D structure that are very interesting when you need to minimize the surface interaction between your droplets and your channels. So it's very interesting for double emulsions, for example. And besides providing this very controlled way of making templates, droplet microfluidics also is very diverse in the types of materials that we could use to make our compartments out of our droplets. So here I have a few examples. For example, lipids to make lipid vesicles, coacervates that are water in water compartmentalization, so without any membranes. And they are thought to be the first type of compartments that we had because they're fairly simple. They're basically the aggregate of molecules with different charges that separate into a polymer rich and polymer non-rich phase. So, and we have also capsules made of polymers. We have nanocapsules surrounding the aqueous phase, and we have proteins. So there's a wide variety of materials that we can use to study the origins of life and use the droplets as templates. I will focus on lipids at this point because they are the most studied ones due to their resemblance to what we know as the basic cell. So before I go into more detail about how we use lipid vesicles in microfluidics, I want to do a quick overview about lipid vesicles. First, they are a self-assembly structure made of amphiphilic lipids. So amphiphilic lipids are lipids that have a polar head and two nonpolar tails. And to minimize the exposure of these nonpolar tails to an aqueous solution, they form a bilayer that put the tails inside, and they self-assemble into spheres, so all the tails are hidden from the outside. Basically, that's the whole concept. So you have an aqueous lumen and also a polar surface and hydrophobic surface, hydrophobic membrane in the middle. The easiest way, let's say not easiest, but the most common way of doing lipid vesicles in the lab is to hydrate a thin lipid film. So you put whatever solution you want to be inside your vesicles in a dried thin lipid film, and with time they will swallow and detach from the substrate. And here you can see that there are several bilayers. So this we call a multilamellar vesicle, but cells, as we know them, only have one bilayer, so they are unilamellar vesicles. And we need to make them unilamellar so we can use them to use them as templates for cells. And we do that by extruding these vesicles through a very thin membrane, and we can control their size when we do that as well. And lipid vesicles can be classified by size. So if they are in the 10 nanometer range, they are called small vesicles or small unilamellar vesicles, SUVs. If they are in the 100 nanometers, they are LUVs or large unilamellar vesicles. And if they are in the micrometer range, they are the giant unilamellar vesicles. And these are the ones that are very interesting for us because they resemble the size of cells. So we'll be talking about these big ones here. And now, how do we make them in microfluidics? There are quite a few ways of doing that. The first one, very simple, is basically you make your droplet. So your lipids are dissolved in your oil phase, and your water phase has whatever is interesting for you to have inside your vesicles. So you make the droplets, and the lipids will act as a surfactant, forming a monolayer around your water droplet. Then you take them and you put them in a solution that has another oil phase with lipids and a water phase with whatever you think is interesting to have outside your vesicles. And by gravity or by centrifugation, depending on the structure that you want, you make your droplets cross this interface. And since the lipids are amphiphilic, they will be right at the interface of these two phases. So once they cross, they form the second bilayer, and then you have your lipids in the bottom of your solution, and you can start using them for your experiments. So this is a very simple way of using microfluidics to make lipid vesicles. The second one is a bit more complicated. So we use double emulsions. So the principle is similar. You do your water in oil droplet, but then you use that solution to have another water layer or aqueous layer outside your oil droplet. So you have your lipids here, and you have to get this oil out so you can have your lipid vesicle. There are a few ways that you can do that, and that depends a little bit on your solutions. So you can tune your solutions to make this process easier, let's say. Here's one example. We have octanol as the disperse phase, as your oil phase. And when you do the double emulsion, the octanol, I'm sorry, spontaneously aggregates in one side of your lipid vesicle and detaches. That process they call de-wetting. I don't like the name very much because it's not wet, but anyways, that's what they call. So the octanol spontaneously leaves your lipid vesicle. So that's one way that you can make lipid vesicles with double emulsions. Here's the schematics, here's the real thing. And this is just an example of using glass capillaries to make double emulsions, and you can see how they look inside the channel, which is pretty cool. The last, but definitely not the least technique, is just my last example. It's inspired in making bubbles with soap. So we'll have here a gap separated by the two supports, and you make your membrane in the middle, and you blow liquid from one side. This liquid can have whatever you think is interesting to be inside your lipid vesicles, and this process makes your vesicles on the outside. They just swallow and detach, very similar to the thin lipid film. It seems very easy and straightforward, but I swear it's not. It's not that simple. Most papers have quite some trouble having a very reproducible production with this method, but I find it very ingenious. That's why I brought it to you. And at this point, you might be asking yourself, so what? So you can make lipid vesicles with microfluidics, and why? Why you can do with them? So let's check a few applications. Here I have two experiments that are very similar. They're from the same group, and what they did, they are trying to study basic molecular, basic metabolic processes of living cells in the bottom-up strategy. So they built the lipid vesicle with microfluidics, and they encapsulated inside it transcription mixture and a RNA detector. So the idea is that as RNA is produced, this detector binds to the RNA and fluoresces. So you can see here that you have 0 minutes and your vesicles don't fluoresce, and as time goes by and RNA is produced by your mixture, you have more and more fluorescence going on, showing that the lipid vesicles could act as a simple minimal cell that can undergo normal biological processes. And the second one is the same principle. They also have the transcription mixture and the fluorophore, but here they encapsulated this mixture and fluorophore inside a conservate and the conservate inside the vesicle to sort of mimic prokaryotic cells. Well, we understood. Basically, so they don't have a defined nucleus with a membrane, but they do have some sort of internal organization, and that's the idea. And you can see here the same principle of the fluorescence being stronger and stronger with time passing and with the production of RNA. So these are two examples of how you can use these structures to study basic symbols, basic concepts. And then the last one, the last example is to use the chip, the microfluidics chip, as a cell on a chip structure. So the chip would be the reaction chamber. And here what they did, they glued sort of DNA to this patch. And this DNA is producing proteins that are fluorescent, and they are trying to understand how the diffusion here happens. So this is sort of how things leave the nucleus to get into the cytoplasm, and to understand the dynamics of the DNA transcription translation. So this is one way of using microfluidics as well that is a bit different from the other ones that I showed you before. And what we are doing in the ProtoMet Consortium is to try to study more complex structures, so hierarchical structures called vasosomes, which are lipids inside lipids, the lipid vesicles inside lipid vesicles. And the idea here is to encapsulate into different compartments prebiotic reaction, so we can see how the vesicles affect the kinetics of this reaction and try to reproduce the first eukaryotic type of cells. And we're going to do that using Elveflow's pressure controller, OB1, connected to our oil phase with our lipids dissolved in them. Then this will be connected to a flow sensor and to the continuous phase of our chip. And then our water phase would have loops. So these ones will be the juice, the giant unilamellar vesicles. And the loops will be the large unilamellar vesicles that might be produced by extrusion. We're thinking about how to do them yet. So we'll have our water phase with our smaller compartments connected to our the dispersed phase. And we'll make droplets and follow the kinetics afterwards. This collaboration is between members of the ProtoMet Consortium, is between Elveflow and ETH Zurich. And I'm very excited to see how these experiments will go. So here I have a few of my reference, or all of my reference actually. If you are curious to look into the studies with a bit more detail, and if you have any questions, please feel free to leave your questions in the comments section. We'll be happy to answer them. And thank you very much.